Hello and welcome to our third video lesson on membrane transport from Chapter 9. In previous lessons from this chapter, we learned how ion transport can work to establish a membrane potential, or difference in charge across the membrane, and how that is used in generating an action potential to send a signal down the length of the nerve axon. Now we want to turn our attention to a consideration of the thermodynamic principles that govern transport, as well as the structure and function of transporter proteins. In this lesson we'll be looking particularly at passive transport and porins. Let's first establish the difference between passive and active transport. In active transport, energy is needed for transport to occur. In other words, it's non-spontaneous. In passive transport, no energy is required because the substance is flowing down its concentration gradient. It's entropically favorable because it's moving from an area of high concentration to low concentration to greater freedom of movement. The simplest example of passive transport is passive diffusion. Some gases can pass freely through the membrane or lipid bilayer, such as oxygen or carbon dioxide, because they're small and nonpolar. That is, they can move freely through that mostly nonpolar lipid bilayer. So this would also apply to other nonpolar molecules, as we'll see. So this is passive diffusion. It is passive in that no energy is required, and it moves through by simple diffusion through that membrane. In other words, no pore or transport protein is needed. In perhaps the most common example of passive transport, we have facilitated diffusion. In this case, transporters pass material into or out of the cell. It is an example of passive transport because no energy is required, but for these molecules they need the transporter to facilitate their movement, and we have that in our figure on the lower right here. Our transporter protein is pictured in yellow, and the substrate molecules that are moving through that transporter are the red spheres. As you can see, we're moving from high to low concentration, so it's spontaneous, but the structure of the molecule is such that it can't pass through simple diffusion through that membrane. Instead, it needs that transporter protein. In other words, it will move down its gradient, but it needs a door through that membrane. Let's look at the thermodynamic expression for membrane transport. In this case, we're talking about passive transport, which occurs when ions and molecules move down the concentration gradient. And we want to look at the free energy of transport into the cell. Let's first of all exclude the fact that there is a membrane potential and look at that free energy change just as related to that difference in concentration, that concentration gradient across the membrane. Here's our mathematical expression. Delta G equals RT times the natural log of the ratio of the concentration of the molecule inside the cell divided by outside the cell. In this case, of course, R is the gas constant and T is the temperature in Kelvin. As you can see from this expression, delta G is negative. In other words, transport is spontaneous or passive only if that ratio of concentrations of our substance in versus out is less than 1 because we're on a log scale, which means that the concentration must be higher outside the cell than inside the cell. But this is what we've already determined conceptually, that the molecule will flow down its concentration gradient, and so in order to flow into the cell, the concentration must be higher outside the cell. But this is our mathematical expression for that concept. So in the absence of other factors, a substance will always flow down that concentration gradient. But in fact, there is a membrane potential, and so to complete our expression for the free energy change of transport, we need to include an expression in our equation. And that's highlighted by the blue oval here. The expression is Z times F times delta psi. Z is the net charge per ion, F is Faraday's constant, and delta psi is the membrane potential. So now our more complete mathematical expression tells us that the concentration gradient alone is not enough to ensure transport. It does depend on that membrane potential. In most cases, however, the concentration difference between outside and inside is so great that the membrane potential contribution will not necessarily reverse the direction of flow, although it can make it more or less favorable. Let's start to look now at those transport proteins, and we're going to start by looking at porins. These are passive transport proteins. They're integral membrane proteins, that is, they pass entirely through the membrane, and indeed they must if we want to move a substance from one side of the membrane to the other. Porins are beta barrels, and that's illustrated at the top of the screen here on the left. In our ribbon diagram, you can see all of those arrows represent the beta strands. These beta barrels have 16 or 18 beta strands, and of course there are loops connecting those beta strands as illustrated here. All known porins are trimers, that is, there are 3 barrels in the functional unit. That seems to be the most stable arrangement and association of these within the membrane. Each barrel functions as a transporter. So within one trimer, we have 3 transporters. The cavity of these porins accommodates molecules up to about 1,000 Daltons or grams per mole, and that's perhaps better illustrated in the ball and stick model on the right. You can see that central pore in each case, a relatively narrow pore, and yet it does accommodate a fairly large size molecule, about 1,000 Daltons. The thing to remember about porins is that they are always open, so they can transport in either direction, and remember that's determined by that concentration gradient and whether the concentration is higher outside or inside. Let's think for a moment about the kind of amino acid side chains that would line the inside of each of those barrels. Of course, it's facing a very aqueous environment, and so we would expect that those side chains would be mostly hydrophilic. Let's look at a good example of a transport porin. This is the outer membrane protein F, or OMP-F barrel, from the bacteria E. coli, and that's illustrated on the lower right here. You can see that very characteristic beta barrel structure in that gray highlighted ribbon diagram. One of the loops connecting the beta strands is very long, and it folds down into the barrel center, and that's highlighted in blue here. That constricts the pore to a diameter of about 7 angstroms, so that nothing's going to get through that's any larger than 600 Daltons. As it turns out, in this one example, the loop has several carboxyl residues, which of course at physiological pH would carry a negative charge. So we say it's weakly selective for cationic substances. Remember, the pore is too large to be restrictive. In other words, even though we have negative charges on that loop, even a molecule that's carrying a negative charge can move through that pore if it's small enough. So it's not highly selective. Most known porins are either non-selective or, in this case, weakly selective. So in our next video lesson, we want to look at transporters that are more selective and see how their structures differ from these non-selective porins.